2026 marks the year cannabis science grows up. No more guesswork. No more strain names with no biochemical meaning. We are entering an era where synthetic biology, enzyme engineering, and microbial biosynthesis collide with cannabis at the molecular level. Today, we're not talking about growing plants. We're talking about programming yeast, redesigning enzymes, and building cannabinoids never seen in nature. This is not hype. This is happening now, proven by peer-reviewed research. And after this shift, the cannabis industry will never be the same again. The old industry is ending. For 60 years, the cannabis industry relied on one biological factory, the trichomes of a flowering plant. But that approach had limits: low yields of rare cannabinoids, inconsistent chemotypes, long growth cycles, environmental sensitivity, inability to scale, minimal control over enzymatic pathways. The science was always constrained by the plant's own biochemistry. But synthetic biology doesn't accept biological limitations, it rewrites them. Cannabinoid. Biosynthesis 101. To understand the revolution, we need to understand the pathway. According to research on olive-tolic acid cyclase, OAC, and tetraketide synthase, TKS, the cannabis plant constructs cannabinoids through a sequence of enzymes. Here is the simplified molecular pathway. Step 1. Hexanoyl-CoA formation. CoA stands for coenzyme A, a molecule that carries fatty acid building blocks. Step 2. Olive-tolic acid formation. TKS plus OAC equals olive-tolic acid. This is the fundamental aromatic backbone of most cannabinoids. The uploaded paper shows that TKS alone cannot produce olive-tolic acid, but requires OAC, a special polykedide cyclase with a unique alpha plus beta barrel structure, to correctly fold the tetraketide intermediate into the final product. Step 3. Granulation to form CBGA, an enzyme called aromatic prenyl transferase. NphB attaches a geranyl tail from geranyl pyrophosphate. This produces CBGA, cannabigerolic acid. CBGA is the mother molecule of THCA, CBDA, CBC. Step 4. Oxidocyclases build the final cannabinoids. Specific oxidocyclase enzymes convert CBGA into THCA synthase to THCA to THC, CBDA synthase to CBDA to CBD, CBCA synthase to CBCA to CBC enzyme. Engineering changes everything. The game-changing moment came when scientists realized we don't need the plant, we only need the enzymes. Breakthrough number 1. Rebuilding the pathway in yeast. Using the complete biosynthesis workflow from the uploaded yeast study, researchers inserted the genes for TKS, OACNphB, THCA synthase into Saccharomyces cerevisiae. Brewer's yeast. The yeast produced cannabinoids identical to plant-derived ones, but faster, cleaner, scalable, programmable, independent of climate or farming land. Breakthrough number 2. Engineering. NphB for new cannabinoids. According to the 2025 enzyme engineering study scientists used, in silico docking, computer simulations of enzyme-substrate interaction, amino acid mutagenesis, swapping building blocks in the enzyme, substrate specificity, tuning to redesign the aromatic prenyl transferase. NphB, the engineered version, can now accept shorter or longer chemical side chains, prenylate new aromatic scaffolds, produce cannabinoids that do not exist in nature. Examples include CBGVA, 3-carbon variant geranyl or selenic acid, 1-carbon variant 2, 4-dihydroxybenzoic derivatives, 0-carbon variant. These completely new molecules cannot be grown in a field, they can only be designed. Breakthrough number 3. Industrial synthetic biology pipelines. Synthetic biology now enables bioreactors producing kilograms of cannabinoids, continuous fermentation, instead of seasonal harvesting, enzyme libraries programmed to evolve new reactions, real-time metabolomics tracking. CRISPR-based pathway optimization. This means product innovation no longer depends on plant breeding. It depends on computational biology. How enzyme engineering works. Let's break enzyme engineering down simply. Enzymes are molecular machines. They fold in three-dimensional shapes that determine what they bind, where they bind, how fast they react, what products they create. If you mutate one amino acid at the active site, you change the machine. The 2025 NphB engineering study demonstrated 4 key methods. 1. Docking simulation. Using Schrödinger software, researchers modelled how each molecule fits into the enzyme pocket. 2. Catalytic distance optimization. They minimized the distance between the reacting carbon atoms, C1 of GPP and C3 of the substrate, critical for SN2-like prenyl transfer. 3. Hydrophobic pocket remodeling mutations like Y288A, G286S, T269. I made the enzyme accept different aliphatic chain lengths. 4. Multi-product screening. Using HPLC-MS, ultra-high performance liquid, chromatography mass spectrometry confirmed. Higher yields, new reaction products increase specificity. The future industry landscape. After 2026, cannabis is no longer an agricultural product. It becomes a biotechnological commodity. Here is how the industry transforms. 1. Pharmaceutical-grade purity. Yeast-derived cannabinoids eliminate pesticides, heavy metals, microbial contamination, variable terpene profiles. Every batch is molecularly identical. 2. New cannabinoids equals new medicine. With engineered enzymes, we can design non-psychoactive anti-seizure cannabinoids, potent anti-inflammatory. Analogues receptor-specific modulators, molecules resistant to oxidation compounds with increased bioavailability. No plant breeder can match this precision. 3. Intellectual property explosion companies patent engineered enzymes, biosynthetic pathways, microbial chassis, cannabinoid analog libraries. The future wealth in cannabis is enzyme IP, not farmland. 4. Collapse of traditional cultivation markets. Outdoor growers cannot compete against 24/7 fermentation-controlled yields, automated purification, synthetic precision. Grow cycles become irrelevant. Final warning and invitation. Synthetic biology is not coming. It's here, and it will reshape medicine, agriculture, manufacturing, pharmacology, global supply chains. Cannabis was just the test case. The winners of the next decade will not be growers, they will be computational enzyme engineers, synthetic biologists, fermentation specialists, bioprocess designers, IP holders. This is biotechnology's next gold rush. The industry will never be the same.